Hey, everybody, what's going on? Welcome into our latest TDR Trade to Black podcast presented by Advanced Nutrients. We've got a new monthly feature we're going to do. It's all about the science of cannabis cultivation. Look, we're heading into a new era, and a lot of companies are realizing that quality is more important than quantity. So we have two very important people, one with a big following, which a lot of people will know here on today's podcast. And we're going to have, like I said, a monthly appearance about this to really educate you on the science behind cannabis cultivation here in 2025, heading into 2026. And I think you'll get some great insights and some feedback based off of this. So this is our latest TDR Trade to Black podcast presented by Advanced Nutrients. Let's bring in Mr. Anthony Varrell. And this is stuff that's right up your alley. You understand this stuff very closely, and should be rather interesting on some of the stuff that we're going to learn with our two guests.
Yeah, I mean, we talk a lot about public policy. We talk a lot about capital markets. And I mean, one of the things that we talk the least about, I would say, was plant science and the actual plants themselves. And there's a lot of cutting edge science and research and technology that are going into everything on the front end, with hopefully we get an increase in research and increase in just actual bandwidth towards studying the plant in itself in a more medical focused scenario, or just really understanding the plant more. And it's going to dovetail right into what Trump was saying a couple of weeks back with the endocannabinoid system. A lot of the work that is going into that will coincide with that as well.
Yeah. So with that, let's get into it. Welcome in two pioneers who help really decode the cannabis cultivation landscape. First, let's welcome in the CEO of Medicinal Genomics and architect of CanMed, Kevin McKernan. He's the CEO of Medicinal Genomics. Good to meet you. How are things?
Excellent. Nice to meet you guys.
Yeah, first time to the podcast as well. Let's bring in architect of CanMed as well, Kevin McKernan. Good to see you. And I think a lot of people know you, Kevin, as well. But gents, you know, we are in towards the back end of 2025. Brendan, I'll begin with you. But what would you say, I guess, are the most important questions that you're being asked in today's landscape when it comes to the cultivation and genomics side of the cannabis industry? Like, what are you hearing most from companies about what they need done or what their concerns are? What can you share with us?
Yeah, no, it's an interesting question, and it certainly evolves as the industry has learned more about the genomics of the plant and how it can be applied to advanced cultivation.
Yep.
So I think there's probably a couple different facets there. You know, as the industry is evolving and moving towards federalization, you know, how do you protect cultivators' novel IP? And that's certainly one application that our company focuses on. You know, we'll sequence a cannabis cultivar. We'll actually take a digest of that and put it into the Bitcoin blockchain to give them immutable records, so they could have a proof of ownership that they had that IP at a certain point in time. We'll then unravel the genome and help them understand what that data means and how do you convert that genetic information into meaningful information to ultimately guide long-term breeding. So how do you build sustainable architecture for agriculture? So those are certainly a couple of components that we work with growers on.
With what you're doing with that, do you see the world of the cannabis industry or cannabis at large or cultivators being able to create defensible moats around their plants moving forward? And if so, how would that work when we've seen such like hybridized strains, and we've seen a lot of people over the years like share genetics? Like how would the moat be efficiently created via what you're doing?
Kevin, I think I'll turn this one over to Kevin here because he has a deeper understanding of plant IP and where genetics can play a role in that.
Yeah, it's a good question, because in this field, it almost seems as if the strains change every 3 years, if not every year. You know, so I think we find people who are doing this only on like mother plants that they want to clone and put a lot of effort into characterizing. And it's usually strains that do something unique, something like THCV or CBDV, and they may not necessarily file for a full-blown patent. They just want to have record of who owned it before they send it off to a nursery, so that if someone in the nursery takes it and runs away with it, they have some legal recourse. Now, Dale Hunt's a good resource for talking about, like, there's a couple different ways you can file for plant IP, and he's probably a better advisor on like what to do based on your business strategy, because there's plant patents that you can obtain, and those are really only good for clonal propagated plants. And they're a little cheaper to file and less intensive. And then there's a USDA path where you actually have to supply your seeds to the USDA. So this one really only works for hemp. But they run it through a dust test, which looks for like distinctness and certain qualities that the plant's actually been bred to be a true cultivar. And you don't have seeds that are all just highly variable when it comes out of it. And then there's utility patents on the plant side. That's usually looking at something that's like molecularly unique, like maybe you GMO'd it or you bred it to do something incredibly unique. And those may remind you of some of the patents that were floating around a few decades ago, maybe 5 years ago, that were a bit controversial when they were going after type 2 plants that were beta-caryophyllene rich. But some people aren't even necessarily engaging in the patent system. They just want a timestamp for, hey, this is when we handed this off to a nursery. You guys cloned it 1,000 times, but don't run off with it. And we have had one case settle, although it settled out of court. It started in court in North Carolina, where they used this sequencing fingerprint to basically demonstrate that, hey, this is the time at which they had the genetics, and this other person who booted up a competitive firm with the same genetics renamed actually had the genetics from his prior employer. And they were able to resolve that dispute based on some etching that we did into Bitcoin. Bitcoin recently had an upgrade as well, which is really helpful because now we can put a little bit more information into the blockchain. Mm-hmm. We try not to put more than like 500 bytes because the community there doesn't like that. They don't like the chain getting bloated with this stuff. But even with 500 bytes, there's a link that I sent you guys that'll show you an example of one from Zamir Punja that we put in there. And I don't think he's pursuing any IP in this or anything. It was just like proof of principle. Can we actually etch the grower's name, the strain name, and a link to the actual genetics and a hash of the sequencing file on chain? And all of that now fits into the Bitcoin op return, so you can actually get nice timestamps that are a little bit more human legible than the ones in the past. In the past, all we have on there was a hash of your sequencing file, and most people in our space, they know what hash is, they just don't know what a Bitcoin hash is.
Right. What is a Bitcoin hash?
So I know what you're saying, Kevin. I've actually had to go in and I went and created a couple of ordinals myself on nodes.
Oh, you have?
On
all right,
the
good.
Bitcoin network. Yeah. So I mean, you're not speaking Spanish to me, but I mean, most people would. Yeah.
So you're speaking Spanish.
Ordinals is another way.
Spanish.
All right.
You're speaking Spanish to me. Maybe explain it.
All right, I'll give you a go on this. So there's a couple different ways to write data into Bitcoin. You can put them in the op return, which recently got a little bit bigger. The ordinal approach puts them in the witness side of the blockchain, which is fine too. It's a different method. That's where like a lot of the taproot institutions have been put into place, which is a new mechanism as of I think it was 2017. But effectively, what a hash is a cryptographic one-way function, which means you take a big file of any size, and it will make a fingerprint, a very long string of numbers that uniquely identifies that file. Now, you can't go from that unique sequence of letters back into the file. You can only go one direction. So every time you take that file and run it through a program like SHA-256, which is the hashing algorithm on Bitcoin, it will always create that fingerprint. If you change one letter in that file, the fingerprint completely changes. So it's really like a small representation of a massive amount of data. So you don't have to put all the data on chain. You just put its fingerprint on chain. Now, you can't go the other direction, which means if you read that hash online or on Bitcoin, you don't know what it means. It doesn't give you any information about the sequence. But you can prove that your genetics existed at the time that block was mined. So it's like a notarization system that doesn't use the federal banking system is a way to think about it. You get a time and no one can change it. It's immutable. And then the benefit now is we can add a little bit more information in there, like a link to Cannapedia and maybe a link to the author. So it's a little bit more human legible now. And I think we're starting to upgrade our systems now to like include that type of metadata into the transaction. So it's not just a cryptic hash.
So it's not so much about creating, let's say, defensible IP or like patents. It's more so creating an immutable catalog of genetics based on the root genomics of the plants at that point in time to create essentially an immutable record of cannabis genetics. So how are growers utilizing that? I mean, because obviously I don't want to distill it down to its elementary form, but we've heard guys be like, well, I was the first guy that was growing runts. I was the first one that was growing jack. Right. Is that basically to create these timestamps with genomic data and be able to be like, I was the first one to grow that, and then all of these clones, clippings came from me?
Yes, you're nailing it on the head, and you can imagine, if you're familiar with ordinals, this is looking deep into the future. You can imagine all types of smart contracts that can be written off this.
Oh, yeah.
Like here's our genetics. We're giving it to this nursery. Clone it, and by the way, if you choose to sell it to somebody else, we want to get a dividend back somehow, and you can pay us.
So
Right.
I don't want to turn it so much into a crypto conversation, but just I'm curious. What was the reason you chose Bitcoin, let's say, over like an Ethereum or a Solana, where Ethereum already has, let's say, tokenization via NFTs, sophisticated smart contracts, and you can transact freely on that platform today with more liquidity and I guess efficiency?
Yeah, so part of that is because we started doing this like 9 years ago, and some of those
Got it. Okay.
platforms weren't really mature yet, and then we just kind of stuck with it. And to be fair, there were times we experimented with other chains because the Bitcoin fees went crazy one time, and during the block wars, and so we did play around with Dash and Ethereum and Bitcoin Cash, but over time we've seen some of those chains have gotten a little bit less reliable or had some changes to them that were like, you know what, Bitcoin seems to be the one that's longest and strongest chain that's surviving the longest, and they started to upgrade their chain to do a little bit more things that you could do like on Ethereum with all the Taproot architecture. So we figured we'll probably just end up back there. The fees are really low right now.
So Brennan, what is your company then? Like Medicinal Genomics? Are you a research lab? Are you a data platform? Are you all the above? Blockchain? Like there's a lot of moving parts here.
It is. It's a great question. I guess it does require a slightly longer answer, but I think if we were to distill it down into like a single sentence, I think we're building the genomic and molecular backbone for cannabis and natural plant industry.
Okay.
We provide the tools, the data, and the scientific standards to help the sector move kind of from, I would say, folklore and marketing claims to measurable biology and defensible IP.
Which is badly needed for this space.
With saying measurable biology, does that infer that your research is enabling cultivators and cannabis companies to, let's say, fine-tune the actual genetics of the plant to how they react with the body moving forward? Or am I kind of reading too much into that?
No, I mean that's an awesome and admirable goal. It's a little hard today. We've been mostly doing this on the plant side because it's a little easier to you can make modifications to the plant or hyperbreed the plant. Can't really do that with humans, unfortunately, right? So it's been a lot. It's been a lot more productive. Like, so one thing we build a lot of these PCR kits that help people look for pathogens like Fusarium and hop latent viroid and Pythium. And those have a really big impact on grows. Like if you get hit with one of those, it can drop your yield 20 to 30%. We make a lot of the cannabis microbial tests in the field as well. So for Aspergillus or total yeast and mold or E. coli, Salmonella. So a lot of there's a lot of labs throughout the country that use our reagents to test for those things. And then the sequencing side, we've been engaged with a lot of different grows that are trying to breed the plant for different capabilities. So there's people that are looking at like THCV genetics, CBDV genetics, and sometimes they're looking at triploids. Sometimes they're looking at there's a host of new tools that have come out very recently in the cannabis space that allow you to breed selectively for different terpene expression. I think the one that's most productive, because breeders, you can't figure this stuff without genetics. People have, right? Genetics just helps move it a little bit quicker. But two areas that I think have been really picking up lately is that people want to be able to move back to seeds. So they want to be able they want a test that can tell them, okay, have I backcrossed this enough such that all my siblings from the seeds are going to be near clonal, right? So you can do sequencing throughout their backcrossing to give them metrics on, okay, you're getting this level of homozygosity, you should be closer to a clonal state. These seeds are stabilized or not stabilized. And then the other aspect is how do we breed for plants that have low total yeast and mold counts? Because that's the number one failure on the microbial side. Is some states with really stringent total yeast and mold tests, the plants are very genetically prone. This is something that Samir Punja has shown that depending on the strain you have, it can change the total yeast and mold counts by an order of magnitude. And it's a very consistent genetic trait. And so when we sequence plants, we try to catalog all the different microbes that are present in the plant, and then also look at the pathogen response genes that are in the plant. So we can try to track, okay, these plants are ones that are going to produce low TYM, and these are the ones that, and here's the actual microbiome that tends to come with the plant.
I mean, big ag has been synonymous with genetically modifying our food.
Yes.
I mean, we've seen it to stress test it, to make it more defensive. To make it, I don't want to say invincible, but give it a very high probability that the farmers are not going to incur crop loss. Do you see the cannabis plant in the future being genetically modified to the extent where it is resistant to mold, mildew, and these sort of outside forces that are incurring crop loss, and that are these grim reapers that are ravaging crops? Or do you think that it's more of a wholesome industry per se with what it's rooted in, and it doesn't want to really take that genetic modification to the extreme to where we're kind of Frankensteining the plants?
Sounds like you need to adapt or die, right?
Yeah, well, I don't know that it's going to happen that quickly. I mean, yeah,
Okay.
there's a lot of things we can take from Syngenta and Monsanto and those parallels, but right now the cannabis industry is semi-decentralized, right? You can't,
Yeah.
unless you're in the hemp industry, which is a whole other shit show. But when you're in the cannabis industry, you can't ship across state lines. Everyone seems to be growing a new strain every 2 or 3 years. That's not
Right.
that's not a very favorable environment to do GMO, because it'll take you that long just to make the change. Now,
Okay.
some people are making some success here. There's a great gentleman who speaks on this at CanMed every year from Wisconsin, where he has managed to GMO plant, and he's done some interesting GMOs. But most of them, you'll notice, we have already seen breeders get to. So, like he's knocked out THC synthase, so we have a really pure CBD plant. But breeders figured that out as well. Very recently, he showed he could swap the terpene genes around, so he could change a plant from being like limonene dominant to linalool dominant, which is actually a pretty impressive feat. He's looking at pathogen response as well at the moment, but we're already seeing people breed for powdery mildew resistance, and there's been maybe 4 or 5 markers that have been identified that you can screen your plants for on that front. So I have not yet seen a GMO in cannabis that does something that breeders haven't done yet. But
Okay.
you might be able to get there faster, that route, if you think the market will tolerate it. But this is a different market. I don't think
Very much so.
the market probably doesn't want that, and
No.
they'll certainly want labeling on it if it's been GMO'd. So it's a much more, I think, complicated market than monoculturing corn, right? Where everyone has to grow the same strain, and how boring would that be in cannabis, right? You go into a dispensary and all you have is Blue Dream.
Correct.
I don't see that happening for a decade in the space, but it doesn't mean the tools people use in the GMO space to monitor genetics and help them breed faster won't still get leveraged in the meantime.
You guys seem like you're light years ahead of the industry right now. I have to ask, when you're all 20 years old, did you think this is what you'd be doing at this stage of your life? Where do you even begin, Brendan, off of something like this? This is highly sophisticated. But like, for example, we had a guest on Monday talking about the hemp industry, and they're looking at some of the labeling and tracking of some of the hemp product that's being sold, and it led them out to a field in the middle of nowhere in the state of Kentucky. So those are obviously big concerns right now, and that continues to happen. But like, where, Brendan, do you even start with these conversations, with some of these companies? And really, what's the ROI they're getting with someone that's dealing with your stack?
Well, it certainly depends on the application. I mean, Kevin mentioned, we work with some growers that are trying to stabilize their seeds, and they're trying to migrate from clones to seeds, and they walked us through their math model. And if we're successful at helping them do this, they can cut their operational costs and their grow by 20%.
Okay.
So the returns are absolutely massive. And obviously, it depends on the application, but that's probably one of the shining areas where genomics can make a very big difference.
Okay.
Outside of that, you have the pathogen component, and that's certainly an emerging area as we begin to sequence the plant and the microbiome. It would be beneficial long term as grows get more sophisticated and they start building quality into the system versus trying to inspect it at the end, which is what traditional manufacturing has done for a long time. So you really want to have an environmental monitoring program where you're swabbing the walls and all of your infrastructure to make sure that you don't in fact introduce a pathogen that could actually destroy crop.
Yeah, like your cocoa. Your cocoa is like your substrate is like the source of most of your problems. And very few people are actually microbial testing their cocoa. They're microbial testing the flower after it's too late and then zapping it with E-beam or something.
Is there a reason they're doing that? Is it a cost thing? Is it an education thing? Is it because I know a lot of growers have their own like processes that they like to stick to and they're finicky. Is there a specific reason for that?
So there's a complicated question in that. Yes, there's some costs involved, but the other issue at hand is that there is there's basically a diversion market here into the hemp industry. Anything that fails ends up there. So there isn't a real high motivation to completely fix this problem, knowing most of this will end up. So, we did a study where we tested 30 samples we obtained online from hemp companies that are, THCA flower type of thing, right? We sequenced them all, their genomes, and they're mostly the same thing labeled differently. And when we order the same strain from the same firm multiple times, it's a different strain, even though it's still labeled the same. Some of them have Delta 8 sprayed on them. They're clearly a hemp plant that someone sprayed, a Delta 8 substance on them. They're loaded with pesticides. They had like a 56% failure rate for aspergillus. That is like tenfold higher than the aspergillus failure rates in the cannabis market, because in the cannabis market, they have to test for it. So I think the hemp market is just the dumping ground. And as long as that dumping ground is open, there is potentially less incentive for people to go in and really address the issues with mold, because they have an out. They can either remediate it and just extract it, or move it into the hemp market. So I think as the hemp market starts to get some regulation tightened around it, I think you've probably seen that letter recently of 39 legislators or something trying to tighten the regulation on this. And this is something we've got to get to Rand Paul about, because he seems to be on the wrong side of this legislation.
Well, he's being lobbied a lot right now, and this is where politics is coming into play. But what were you going to say, Anthony?
I was going to say, he very much so is on the wrong side of this. I mean, if you listen to our interview that we had with him about 6 weeks ago, he kept claiming that the culprit is Delta 8 THC. He didn't want to talk about synthetics, THCA flower, and just the complete inversion that's happening on the THCA flower market coast to coast. But I mean, with what you guys are doing, messing around with blockchain 9 years ago, and where you are in the industry, have you had any talks with state or federal level officials about putting the entire industry on the blockchain once and for all? Because it would solve inversion issues. It would also be able to create a more immutable framework from seed to sale, and it would help fix a lot of our problems, especially if there was congruency that was achieved between hemp and cannabis once and for all, and it was just regulating THC for the most part.
So I don't have great inroads to those sources. I may run into Rand Paul next week at a conference in Austin, which I'll try and bend his ear on this about.
Love to hear how that goes.
Yeah. I mean, I respect what he's doing in a lot of other areas. I think in this area, him being from Kentucky, he's probably just given the wrong information. He's probably thinks he's saving Kentucky growers, and it's actually inducing a THCP market and a bunch of other synthetics, and we're even seeing kratom compounds on these things, which is nuts.
Yeah.
And that stuff can be addictive. Not necessarily the amounts in kratom, but if you synthesize 7-hydroxy at high concentrations, that's definitely an opiate. But the non-hydroxylated version of it seems like it's a much safer option that's more prevalent in the plant. But yes, I think he's been given bad information to protect the Kentucky market, and those people aren't growing stalks and seeds. They are growing clearly cannabis that is being sold into gas stations and smoke shops, and it's completely untested and unregulated. And what I don't like about that, I know it sounds like we're shilling for our own tests here, but I've got multiple friends in like my neighborhood whose kids have gone into these gas stations and gotten pens and gone and freaked out and gone to the ER. So, and then they think
you've seen that firsthand. You've seen that firsthand?
I've seen that firsthand. Absolutely. Yeah. And so all of my friends are like, what the hell are you doing in this industry? Our kids are getting these pens now. And I try to explain to them, it's not the cannabis market. This is the hemp market. And they're like, isn't it the same plant? Like, yes, it is. But, and so I think we have to regulate it that way. There is some motion getting behind this from WANA. And I think there's a conference on this in Vegas in early December. To try to regulate this as one plant. Like, let's cut the bullshit here. This is the same species, and let's come up with some regulations that make sense for both. So we don't have this arbitrage in place where you just put the hemp hat on and suddenly no rules apply.
Yeah, I get it. Brandon, let's honestly, when you're working with companies, like, where does the conversation begin? This is very high level. It's very sophisticated, but it is a solution to Anthony's point. But where does the conversation begin for those that are watching this for the first time and want to learn more?
Well, depending on the business area of interest or the challenge that they're trying to deal with. If it's microbes, it's relatively easy. We have molecular tools that can help them, analyze, what's growing in their facility, what's growing on their coker, on the plant, on the flower. And Kevin's team has really boiled the technology down to make it easy enough that a high school student could run it. And that was one of the bottlenecks we've had in the past. Our first generation platform was really easy for a laboratory to operate, but you needed to have a technical staff to run the platform and understand microbiology. That wasn't easily portable to a cultivation facility unless they hired the right staff. So Kevin's team has re-engineered it, integrated to a really simple automation platform. So you could hire anyone in the world to actually run the platform and get sequencing data.
Turnkey.
Data.
How much is AI and machine learning accelerating what you guys are doing? And do you think that there's going to become a day, let's say, do you think that there's going to become a day in the future?
Every day.
Okay. Do you think there's going to become a day in the future where indoor cannabis facilities look more like dark factories, where it's all automated, there's no human interaction, and they can create these contained environments to go?
I think
you're seeing that now, are you not?
Absolutely. And without a doubt, we're already using it now just for, there's a host of things you can use. It's actually really good at genetic information. Like ChatGPT 5.0 is very good at handling sequence information. You can ask it to run a lot of your bioinformatics pipelines. You can even give it a whole cannabis genome and say, tell me which variants in here are like most interesting for yield or growth. And it will come back with a long list after combing through it for a few minutes of, hey, these are some important variants to track. So I'm really impressed with what it can do. Yeah, you do have to spot check it, but now 90% of your time is freed up, and you only need to spend 10 minutes, spot checking it. It's very. I think it's going to really change the way we make use, at least, of our data. On the growing side, we don't operate a grow, so I don't want to speak for where it's going to impact them the most. But I would imagine there's certainly some robotics that could be managed by it to help with. Intelligently going through the grow and looking for problems and correcting them before they happen.
Yeah. In some cases, unfortunately, we're contacted too late where the house is on fire and they need a team to come in and help them figure out what is the pathogen that's destroying our facility.
Okay.
So it's all part of trial and error. It's all part of the progress, right? But that's where we are headed, right?
Yeah, that's where we're headed. You look at traditional crops; it's done every day. Over time, they start using genetics to kind of, as Kevin mentioned, they'll modify the plant to optimize which ones have better disease resistance and integrate that into plants that are weaker but it has better phenotypes. And we'll see that emerge over time. You'll start mixing, and breeders do that naturally. They find a plant that's powder mildew resistant, it doesn't necessarily have all the traits, and they start breeding it with other plants to try to marry the two together. We can just do that a little bit faster just because we have intelligence.
I asked a question earlier. It was kind of a runoff question, but again, I'll ask you, what is your background, Brendan and Kevin? Like, Brendan, where did you, I guess, go to school for this, and how you got involved in this whole career and this idea?
Well, Kevin's a scientist of the group. I know enough to be dangerous.
Okay.
But our family's been in the life science business for decades, and it actually dates back to the Human Genome Project. Kevin worked for Eric Lander to build the first pipeline for the Human Genome Project. I joined him for a very short stint, but during that process, Kevin patented a novel chemistry called SPRY. And it was a way to automate the process of extracting RNA and DNA in a highly automated fashion from the genome. So our family actually came together and we licensed that technology from MIT, and we formed our first fully family venture called Agincourt Bioscience Company. And we became one of the world's largest genomic services companies. I think we were the only private entity to be selected by NHGRI to sequence high priority genomes for the government. And that was for animals, that was for humans. I think we even sequenced all the microbes on the Mars rover. And eventually that company was acquired by Beckman Coulter. And then Kevin went on to invent a novel sequencer that cut the time and cost of sequencing by a factor of 100,000.
Wow.
That company was quickly acquired by Applied Biosystems and LifeTech. And Kevin kind of went with that transaction and ran worldwide sequencing for LifeTech. My older brother and I left, and we actually wanted to move next-gen sequencing into the clinic to help kids with epilepsy, autism, and migraine. And that's where we saw the overlap with cannabis.
I was going to say, and that which led you to cannabis, right?
Which I never thought I'd be in this space. At that same time, Kevin left LifeTech, and he actually formed MGC back, I think, in 2011 to sequence the first cannabis genome. And we lobbied with the board to acquire Kevin's company and bring the technology in house. And that's how we got into the sector.
Yeah.
So we didn't attack it from the plant. We actually came through really a different path of building a lot of the innovative tools that the sequencing industry uses today.
What's the most exciting thing, I guess, Kevin or Brennan, that you're looking at right now in plant science or in genomics that you think we'll be talking about, let's say, at CanMed 2026 in June of next year?
Yeah, right now we're playing around. I should have brought one in here. We're playing around with these little like nanopore sequencers that are like smaller than your phone. And they've now gotten to the point where they're accurate enough that you can sequence like a microbe in 10 minutes. Like it's phenomenal. Okay. And so now if you need a question, like what we have people do is they send us a colony on a plate that they're concerned, like this came off my plant. I don't know what it is. What is it? They can ship it to us and we can get that genome sequenced and assembled into one piece in pretty much 24 hours and tell them, all right, this is Salmonella or this is E. coli or this is Pythium. We couldn't do that for the vast majority of our career. We'd have to send it on, put it on an Illumina sequencer, which would take a week, and then analyze it for a week. And now the genomics are so fast that you can get really rapid turnaround time on figuring out what microbes infesting your facility. So that's been really fun because we've never seen things just hustle through the lab that quick and turn around as fast as they have. And the machines they're $5,000. I mean, this is like an incredibly low barrier to entry. The sequencers we were building a decade ago were like $500,000.
Yeah, I was going to say, why are they only $5,000? That's cheap.
It's just a breakthrough on how they do it with these nanopore sequencers. They're really small and then they're portable. So I can sequence some. I've sequenced stuff on my kitchen table. Like I'll just bring my laptop home with a sequencer, and on the weekend I'll throw shit on there, and I'll have a genome by Sunday. So that's going to change the way we think about pathogen surveillance in the future. Now it's probably the reagents to run it are like, it's about $1,000 to run the machine, which is a lot higher than PCR. So, this is great for genomes, but it's not the fastest, cheapest way to actually like qPCR something. We can qPCR something in a couple hours and it's probably, $10. It's not $1,000 to run. But as those prices come down, we'll start to see that as a tool that will decentralize maybe even into the grows, where they can be looking at the stuff in site if they need to.
I was going to say,
that might be 5 years away, but we'll see.
So this process, the way it is right now, this process is more reactive versus proactive. But as you get the cost to come down and the efficiencies to come down, they can start using it in a proactive sense as the cost would make sense.
Yes. Yeah. Okay. You could start swabbing your facility. And if you have anything, I think what most people do is screen with PCR to see if there's anything there. And if there is, throw it on a sequencer and figure out what it is that day. And that kind of rapid turnaround time will make it really responsive for these grows. As things go to into an FDA environment, which I hope is a long way off because the FDA's got a lot of problems and they're very centralized. Yeah. But if you think about their mantra in terms of being proactive on this, which is what they do in the food industry, is they're constantly surveilling their surfaces to test for Salmonella, Listeria, and E. coli. And whenever they find a hit, the FDA always encourages people to like proactively figure out whether it's a resident microbe, something that's been in your facility before, or if it's a new one. Because there's different things that you do if it's resident versus novel.
Easier said than done, though, right?
Yeah, it is. It does require some proactive screening efforts, but this would ultimately be in an IPM program, right? I mean,
I look at this and I think this industry, what's the biggest challenge they always face is consistency. If they want to have a proper CPG industry, like we've been to various facilities, I won't say names, but Anthony, I've seen where plants are 5 feet tall, another one's 2 feet tall. There's no consistency as far as height when looking at the overall cultivation crop as to what they're growing, but that's always going to be a challenge. And I guess in a lot of ways, is that some of the questions you look to solve among other things that you've already outlined here today? Question for either one of you.
Oh, yes, certainly. And I know exactly what you're talking about. I've been to a couple of grows up in the Sun Belt, actually, where they grow sticks, short and just one large cola and lots of tightly packed pots. And that works for them. And they're not ashamed to call it Bud Light. It's like they're going for the high volume market that wants a small flower size that doesn't jam up the trimmers. And there's others that want to grow like the best nugs in the world and have really good bag appeal, and they're not interested in the pre-roll market. So they grow different plants. I think that's what's exciting about the cannabis field is I don't think there is one answer for how to do this right. It's like we already have a microbrewing industry immersed inside of a Bud Light industry. Both of these markets are here. And there's going to be room for people who are making really high quality cultivars that have unique terpene profiles and unique cannabinoid profiles. And then there's going to be, like 80% of the market's just going to want, gummy bears, and they don't care where it came from.
But whatever lane you're in, you got to be consistent is where I'm going with this,
true.
right?
Yes. Yeah. And those tools matter for both markets. And that was kind of the point I was trying to raise, is that consistency is going to be important whether you're in the microbrewery or whether you're in Bud Light.
Yeah, makes sense. And I was going to ask you a question. Oh, sorry, you want to add something, Brendan?
Yeah, no, I was going to mention, and this kind of goes back to something that Anthony brought up. One of the elements that we're actually covering at next year's CanMed event is a global policy, a global standards one plant policy forum, which will actually go into what's the federal playbook. And we're bringing in folks from the USDA and from the food industry and the nutraceutical industry in terms of how would cannabis get managed underneath that framework.
Okay.
And that will roll into discussions on sort of rooms to zones. How does the food industry manage pathogens?
Right.
What are the best practices to make sure that you have standards and consistency to make sure consumers don't get sick? So we're going to cover a lot of angles at CanMed this year from orthogonal industries on what our sector can actually learn from others.
Not to get too elementary, Kevin, but the age-old question: hybrid, sativa, indica. How much of mapping the genome and the work that you're doing settles that debate?
It doesn't
once and for all.
Yeah, I wish it did. So there was a paper from Hazenkamp actually in Holland. I don't know if it made it to a peer review paper, but I remember seeing it at an ICRS conference where there was some meaning in the Holland market in Netherlands of those terms, and I think it may date back to the fact that they had more segregated genetics 20 or 30 years ago. But now in the States, when we try to look at the cultivars here, it's all mixed. Like when we look at them genetically, those names don't really mean anything to us, unfortunately. And I know a lot of dispensaries like to sell on indica and sativa, and
it's like 95% hybrid.
Yeah,
now.
And I can't justify it. Maybe one day it'll all make sense to us, but it doesn't line up with the genetics. The nomenclature doesn't line up with the genetics. Now, part of that may have ultimately get solved if we do start recording this stuff in immutable ledgers. Because I think some of this is really word of mouth tracking of these genetics. We got people writing Sharpies on bags, and God knows what the fidelity of this is in tracking these things. So we may be able to get there one day, but at the moment, it's still it doesn't correlate well with the hybrid sativa thing. Doesn't really correlate very well. The leaf structure does. We have seen some genetics that imply the sort of the wide leaf and narrow leaf types are in fact do cluster on the phylogenetic trees. And the hemp plants versus the type 1 cannabis plants do cluster in different areas on the phylogenetic trees. So there is it does make some sense. It's just not resolving that particular issue.
Okay. June 15th to the 18th, 2026 in Lake Tahoe. That is the next CanMed event. Maybe explain to us a little bit. You've had this event last year. It was in Puerto Rico, correct?
Correct.
So who are some of the people, notable names that attended last year?
We get leading. It's an interesting intersection of the community because you get some of the leading scientists. You have physicians. You have cultivars there, along with entrepreneurs just building businesses. So it's a really interesting mix.
Like, are you getting the Cure Leaves and True Leaves and that type of the world that are attending these events? Yeah. Okay.
Yep. True Leaf has been there for a number of years. They're really fascinated by the medical applications of cannabis, and they've built a whole medical team. In fact, they send their chief medical officer attends CanMed to go to the medical practicum to learn about dosing, toxicology, clinical cases. How do you apply cannabis to help human health? And then other grows come, even here locally in Massachusetts, we work very closely with Happy Valley. They send their head of cultivation here to learn about the leading edge in genomics. We have a whole workshop focused on the use of genomics and CRISPR and gene editing technology and how that might be applicable to their grow. So lots of different topics covered, but it's a very deep technical conference.
On the medical side, some of the names you might recognize are Bonnie Goldstein and Dustin Sulak and
Ethan Russo this year will be in the medical practicum.
Okay.
Deddy Mieri's a really good one. I always like his talks because he's taking these cannabinoids and putting them across cancer cells and seeing some really fascinating results.
Wow.
There was just a phase 2 study, I think that was announced today coming out of the UK with
Oh, really?
using THC and CBD in Alzheimer's.
Oh, I
And,
did. I
and
saw that.
dementia.
Yeah,
Yeah.
that's needed actually. That is on
Would
the
love.
rise, and
Do you think, I don't want to get too charged here, but I do see you on Twitter, Kevin, go and kind of put the Berensons of the world in their place
yes,
when it comes to the
it's not very
misinformation
hard
and
to
just
do, by the way.
correct and the debunking of their studies that are completely uncontrolled and just pseudoscience
Yeah.
to that degree. Do you see us in the next couple of years getting to an inflection point where these trials that are using cannabinoids for actual medical applications outweigh these just reefer madness pseudoscience studies that really have no place in conversation?
I am a little bit less optimistic that that's going to overturn it. I think what we have to do as a community is fix the supply chain, because all of these papers are getting confounded by the fact that we don't know what they're consuming.
Correct.
Right? Like they'll do a study on like cannabis users and non-users and declare like one shrinks your testes or something, right? But you don't know if they're smoking hemp that's loaded with pesticides, or what it is, and that we know from looking at a lot of the papers. I think the Gagnon paper in Canada was really interesting because they looked at the black market versus the tested market after legalization, and the black market had like 92% of the samples were pesticide loaded, and only 6% of the legal stuff had pesticide contamination. So there's a massive difference in pesticides, and the pesticides are probably what's causing the psychosis, right?
Yeah.
These are all neurotoxins, and they enrich in extraction, and some of them make hydrogen cyanide. I mean, this shit can be really, really dangerous. And then you have Berenson running around being like, the weed caused psychosis. It's like, you have no idea what they consumed. Right? And if there's myclobutanol in there, I guarantee you that's much more dangerous for psychosis than THC. And so we have to fix that in the community. Otherwise, all the papers are going to be based on that kind of trash association where we're just saying users versus non-users. If we don't know what it is they're consuming, like you take schizophrenia, they're talking about using CBD to treat it, right?
Yeah.
So like if you don't know the chemotype of the plant of the user, how are you ever going to make sense of a study like that? Some people could be using 1-to-1 strains and they may be preventing schizophrenia.
Yeah.
So
I already know the next conversation I want to have with you guys, and we'll have this next couple of weeks. But if rescheduling were to be announced, yes, we look at the actual headline itself, but then from the FDA, all the requirements that are going to be asked and what's going to happen after that, I think we really need to dig in deep from a science perspective, because rescheduling is not anything to do with recreational. That's obviously on research, the medical side. So it'd be really good to learn more about if that announcement were to happen. And I don't want to get too much into it because we could be here for another hour talking about that. I want to turn that up.
Yeah.
But
I would at some point love to pick your brain on that, because I think you guys have had some great shows discussing the implications of Schedule 3, and it does seem like it may change banking, which could be significant.
Would be massive.
Yeah.
It's just got to happen. But you need to speak to people like Senator Rand Paul and make sure that you're educating him on really what's at stake, because this hemp side is gaining some momentum for not the right reasons. And look, Anthony and I have always been very, very transparent that we don't want hemp basically to falter. It's just getting rid of the bad actors, and that's important right now. So
yeah, I'm with you there. Like we want it all to be treated the same, so there isn't this crazy arbitrage, and we're all for legalization.
Yes.
It's just what's happening now is I think it's creating a massive economic incentive to do the wrong thing. And then all the adverse events of that wrong thing are getting blamed on people trying to do the right thing.
Agreed. Yeah. They're not thinking these things through. Senator Mitch McConnell did not think things through when it came to the Farm Bill in 2018, and I think he could be making the same mistake here 7 years later.
If you're a prohibitionist and you wanted to create an existential threat on the state-run cannabis industry or the legal market, you would have created the Frankenstein hemp market to unlawfully just wreak havoc. And like Kevin alluded to in the Berenson studies, they're not saying the kid smoked a THCP or an HHC vape that was loaded with free radicals and heavy metals. They're just saying cannabis. Yeah. It's the perfect storm if you wanted to take down the legal industry in a very indirect way.
Bingo.
But we're putting a lid on it, hopefully day by day, and getting progress to get those bad actors out of here.
Let's walk before we run. Brandon McKernan, CEO of Medicinal Genomics, along with the architect of CanMed, which is taking place June 15th to the 18th in Lake Tahoe, 2026. Here's a crazy question that I should probably know. Lake Tahoe is in Nevada, right? Not California.
It's on the border, isn't it?
It's on the border, but the resort we're staying at is on the Nevada side.
All right. We've spoken to the Advanced Nutrients team, so it sounds like we might be going to that. So that'll be very, very exciting.
Awesome.
But
Yeah, so 10th anniversary, so it's going to be a great one.
Oh, so this has been going on for 10 years.
Yeah,
Yeah.
Raphael
Yeah,
McKernan actually helped us boot it up back in Harvard Med School.
Wow. Okay.
Yeah, he didn't see a pure play scientific conference that was data driven and free from commercial influence. So
Okay.
His goal was to help as many patients as possible, and he thought a lot of the conferences in this space were really focused on the commercial side.
They were.
We need to we need to get some lawmakers at that conference next year.
Yes.
But, that would be very.
Well, we invite politicians every single year, and they turn us down every single year.
Why do you?
Now we do get regulators there, but it's not the same thing. The folks that are managing the legal cannabis market versus
Get the decision makers, not the gatekeepers. Kevin McKernan, genomic scientist, along with the CEO of Medicinal Genomics, Brendan McKernan. Again, gentlemen, thank you for your time, and look forward to more of these conversations. Just to tee everybody up, next conversation with these guys is going to be about if rescheduling is announced, then what happens after that from a scientific standpoint, because a lot of questions are going to be asked by the FDA. So we'll have that coming up in our next segment here with both Kevin and Brendan. AV, thanks for joining us, everybody. Enjoy your day, and we'll see you guys next time. This is latest TDR Trade to Black podcast presented by Advanced Nutrients.
All right, thank you.
Thanks.
Hey everyone, thanks again for watching here today. Listen, if you're getting value from us here at TDR, make sure to check us out at TDR Plus. It's our exclusive membership community. Why? Well, you'll see CEO interviews that go beyond business, showing a personal side that you won't find anywhere else, including the biggest names in the industry. Plus, you'll get insights from analysts and day traders on where the industry's headed, both short-term and long-term, and a direct chat group with our team. It's only $7 a month, just 25 cents a day, and it helps support everything we do here at TDR. If you want to join, just click the join button below and become a TDR Plus member today. And once again, a big thank you for being part of our community. So that's it for today. And again, thanks for watching, and I'll see you next time.